friends, myself Shoptapurni from Biolive. Today we will continue the intrinsic pathway of apoptosis. Okay, let's begin. See, whenever there is any kind of damage inside our cell, okay, like it could be a DNA damage or it could be like any kind of stress or trauma or any kind of like lack of oxygen supply or lack of nutrient supply in our cells, then an intrinsic signal of death comes from inside of the cell, okay, and then what kind of pathway appears? That pathway is named as intrinsic pathway of apoptosis. So let's write intrinsic pathway of apoptosis, okay. So here the death signal arises inside from the apoptotic or target cell, okay. So what happens here? Let's say this is our target cell, okay. This is our target cell. Inside our target cell, there is mitochondria. And in the intermembranous part of the mitochondria, cytochrome C is present, okay. So, when an apoptotic signal comes from inside the cell, then this cytochrome C gets transferred from the intermembranous part of the mitochondria to the cytosol. Okay, so now the cytochrome C are here in the cytosol. When they reach cytosol, what happens? Inside the cytosol, there is a protein, adapter protein, named as APOF1. This is the APOF1 adapter protein, okay. Cytochrome C comes and with the expense of one molecule of ATP, it binds with the APOF1. How? See, it binds like that. So, this is the cytochrome C and this is the APOF1, okay. And this is the card domain of APOF1. This green one is the card domain of APOF1. In the next step, what happens? 7 APOF1 binding with the cytochrome C arranges themselves in a wheel-like manner, okay, or wheel-like arrangement. And that arrangement, that heptameric arrangement is known as apoptosome, okay, apoptosome. So now what happens? Let me draw 7 APOF1 here. Okay. These are the cytochrome C binding with the APOF1s and these are their card domain. With the help of card domain, they bind with each other. Okay. Now, this is the apoptosome. Apoptosome. In case of intrinsic pathway, the initiator procaspase is procaspase 9. So, inside the cytosol, procaspase 9 is also present, which look like this, which has also a card domain. This blue one, this green one is the card domain, and this is the procaspase initiator procaspase 9. Okay. Now this initiator procaspase 9, 7 initiator procaspase 9 with the help of their card domain binds with the card domain of APOF1 and form another structure. Okay. See, like this. These are the card domains of our procaspase 9. This is the prodomain and this is the procaspase 9 structure. Okay, so like this, 7 procaspase 9 bind here with the apoptosis. Okay, now you can see that now the procaspase 9s are in close proximity. So we have learned in our previous lesson that whenever the initiator procaspases are in close proximity, they cleave each other. Okay, so now see if take these two procaspases, okay, initiator procaspases, so they will cleave each other at their cleavage sites, and as a result they will form this kind of structure, the active tetramer or the active initiator caspase. Okay, active initiator caspase 9. Okay, this is the tetramer. Now this active initiator caspase will activate other executioner procaspases to their executioner caspase form, and they will activate the caspase cascade, and will also cleave the target proteins like nuclear lamins, cytoskeletal materials, the inhibitors of endonucleases, etc. Okay, and then the cell will be apoptosized like this. Okay, the cell will be apoptosized and cell will form blebs or bulges, this kind of bulges. Okay, cell will form this kind of bulges, and then these bulges will detach from each other and they will known as apoptotic bodies, and they will be engulfed by the macrophages, and that is how the intrinsic pathway will complete. So this is the intrinsic pathway of apoptosis. Now one thing is important here. Why the cytochrome C is released from the intermembranous space of the mitochondria to the cytosol? What signal causes their release, or what regulates their release? Okay, now let's talk about that. In our body, there is a regulated protein, family of protein actually, known as BCL2 family of proteins, okay, which regulate the intrinsic pathway of apoptosis. So, regulator protein family is BCL2 family of proteins. Okay, family of proteins. We can divide this BCL2 family of proteins into 3 types. What are those? Number 1, anti-apoptotic protein. Okay, anti-apoptotic protein. That means those BCL2 family of proteins which inhibit the apoptosis. What are their examples? BCL2 itself and BCL-XL. Okay. Now, second one is proapoptotic protein. Proapoptotic protein of BH123 family. Okay. Or BH123 homology. What are the examples? Bax and back. Bax and back. Remember, Bax and back are the BH123 family of proapoptotic proteins. Proapoptotic protein means those proteins which activates the apoptosis or stimulates the apoptosis. Another type of proapoptotic proteins are BH3 family of proteins under the BCL2 family. And what are their examples? BEAD, DAD, BEAM, PUMA, NOXA. Okay. So remember, these are the 3 family of proteins under the BCL2 family. Okay. All of them are regulated proteins of the intrinsic pathway of apoptosis. Now, why these names? BH123, BH3? Because all of these proteins, the BCL2 family proteins, has 4 homology domain, okay, known as BH4, BH3, BH1 and BH2 domain. The anti-apoptotic proteins share all 4 domains among them, but the proapoptotic BH1 to 3 proteins share only these 3 homology domains, 1, 2 and 3 domain, okay, and the BH3 domain has only this BH3 portion of the BCL2 family. So that is why they are named as such. Now, in our body, there is a balance, always there is a balance between these pro and anti-apoptotic proteins. Whenever a cell is in normal state, what happens? There is a balance, always there is a balance between pro and anti-apoptotic proteins. When the apoptotic signal comes, the pro-apoptotic proteins gets dominant and anti-apoptotic proteins gets repressed, and as a result it causes apoptosis, okay. So now coming to their function, or how do they regulate. See, we have learned that in the intrinsic pathway, if it is our mitochondria, it is the outer membrane of the mitochondria, and let's say this is the inner membrane of the mitochondria. So this space is the intermembranous space, okay, and this is the matrix inside the mitochondria. Let's draw it ring, okay. So in this intermembranous space, we know that cytochrome C are present here. Now, when apoptotic signal comes, this cytochrome C is excreted out or transferred to the cytosol. But how are they transferred to the cytosol? Actually, what happens? On the outer membrane of the mitochondria, BH1 to 3 family of proteins, that means backs and back, okay, they form a oligomer like this. That means if it forms, see, this is a backs protein, this is a backs protein, so they form a channel like this, okay, arranging like this. Through this channel, the cytochrome C can go from inside to outside of the mitochondria. But in a normal cell, non-apoptotic cell, this oligomerization does not form, okay. What happens? We are now talking about BH1 to 3 family of proteins under which backs and back come, okay. In a non-apoptotic cell, this back is always present on the outer membrane of the mitochondria, but they do not oligomerize to form a channel. That is why cytochrome C cannot go out, okay, and backs is present in the cytosol like this. Whenever a apoptotic signal comes, this back oligomerizes, or this backs comes to the outer membrane and also oligomerizes to form a channel, and through which the cytochrome C then can go out, okay. So now one question may arise: during the normal state of the cell, what inhibits them to form a channel like this? They are inhibited by the anti-apoptotic proteins, okay. What are those? BCL2 family of proteins like BCL2 and BCLXL. So anti-apoptotic proteins in a normal cell, okay, actually inhibits them. That is why they cannot form a channel like this. What happens? Whenever an apoptotic signal comes, then you need to activate them to form the channel. For that, you need to inactivate this anti-apoptotic protein. Then only they can form the channel. So what inactivates these anti-apoptotic proteins? The BH3 family of proteins. Okay, that means what are the BH3 family of proteins? Bead, beam, bad, puma, noxa. Okay, these are the anti-proapoptotic proteins of BH3 family. They actually inhibit these anti-apoptotic proteins. So as they are inhibited, now they are activated, they can form the channel, and through which now cytochrome C can go up. Or they can directly activate these BH123 family of proteins to form the channel. Okay, that is how the intrinsic pathway is regulated by the BCL2 family of proteins. So, in a gist, we can write that the BH123 family of proteins, that means BAX and BAG, when oligomerizes, they form a channel through which cytochrome C can go out. Okay, but these BH123 proteins are actually inhibited by anti-apoptotic proteins like BCL2, BCLXL, etc. So to activate them for oligomerization, you need to inactivate this anti-apoptotic protein. That will be done by BH3 family of proteins. Okay, if they inactivate them, then automatically it will be activated to form oligomer. Or they can either directly activate them to form the oligomer. What are the BH3 proteins? Puma, noxa, bad, beam, bead, etc. Okay. Different BH3 proteins acts at different apoptotic signal. Like puma and noxa works when there is any kind of unrepaired DNA damage in the cell, and then the apoptosis is needed, then the puma and noxa are activated by the P53. Okay, the tumor suppressor gene of our body. Now, there is a link between extrinsic and intrinsic pathway of our body. Okay, whenever an extrinsic pathway is activated, our body tries to amplify that death signal by activating the intrinsic pathway, and that is done by the bead protein, the BH3 family protein bead. What happens in the extrinsic pathway, we know that procaspase 8 and 10 gets activated to caspase 8 and 10. Okay, so this caspase 8 cleaves the bead into T bead. Okay, T bead. This T bead then goes to the mitochondria and it activates the proapoptotic proteins of BH123 family by inhibiting BCL2 or BCLXL or anti-apoptotic proteins. Okay, anti-apoptotic proteins, and that is how it activates the extrinsic pathway, activates the intrinsic pathway of apoptosis. Got it? And that is how the death signal is amplified in our body. Now, besides this BCL2 family of proteins, there are other types of regulated proteins in our body. Those are known as IAPs. Okay, IAPs. That means inhibitor of apoptosis. They are mainly first found in the Drosophila body. Whenever a Drosophila is affected by baculoviruses, baculovirus tries not to kill its host cell, okay? That is why they form this IAP or inhibitor of apoptosis. This IAP, what causes it? It binds to the caspase 3, okay? Actually what happens whenever in the intrinsic and extrinsic pathway, the main executioner caspase which is activated is caspase 3, and that caspase 3 causes the cleavage of all target proteins. So this IAPs binds to the, let's say this is the caspase 3. So IAPs and this is the IAPs, okay? So IAPs binds to the caspase 3 and blocks it, and that is how they inactivates the apoptosis pathway. There is also anti-IAPs inside the intermembranous part of the mitochondria. They actually binds to the, if this is the IAP, the anti-IAPs, and if this is the anti-IAP, anti-IAPs binds to the IAPs, okay, and they inactivate the IAPs, so the caspase 3 remains free and they can then cause the apoptosis. Got it? Anti-IAPs in our mammalian body is known as MAC or Diablo, OMI, etc. Okay, and in Drosophila body they are known as Reaper, Grid, etc. Okay. So from this we have learned that in our body, intrinsic, there is a balance between BCL2 family of proteins, the proapoptotic and antiapoptotic proteins, and also the IAPs and anti-IAPs, so that when the apoptosis should occur or should not occur can be regulated in a controlled manner. Okay. So with this, we come to an end of our today's lecture here. If you like my content, please like, share and subscribe to my channel, and also comment below. Thanks for watching and happy learning.